Greetings to all the learners. Today we are going to learn on the topic Holliday model of homologous recombination. To begin with, the Holliday model was discovered by the scientist Robin Holliday in 1964 in E. coli. Homologous recombination exchanges DNA between homologous chromosomes during meiosis, producing genetic variation and DNA repair. The functions include genetic variation, DNA damage repair, replication restart, and regulation of gene expression. The key steps involved in Holliday model of homologous recombination includes alignment of homologous DNA, double-strand break and processing, strand invasion, Holliday junction formation, branch migration, and resolution. In this slide, you can see the picture of duplex DNA and heteroduplex DNA for your reference. The first step is alignment of two homologous DNA molecules. Homologous refers to the DNA sequences which are identical or nearly identical for a region of at least 100 base pairs. After the alignment of the homologous chromosome, introduction of breaks in the DNA occurs, what we call it as nick formation. The enzyme endonuclease cuts single strand of each homologous DNA at the same position. Once the breaks are formed, the ends at the breaks are further processed to generate regions of single-stranded DNA. Strand invasion. The single-stranded DNA tails generated by this process then invade the unbroken homologous DNA duplex. Initial short regions of base pairing are formed between the two recombining DNA molecules. This event is called strand invasion. Formation of Holliday junction. After strand invasion, the two DNA molecules become connected by crossing DNA strands to form a structure that is called a Holliday junction. This junction can move along the DNA by the repeated melting and formation of base pairs. Each time the junction moves, base pairs are broken in the parental DNA molecules, where identical base pairs are formed in the recombination intermediate. This process is called branch migration. Resolution of the Holliday junction. The process to regenerate DNA molecules and therefore finish genetic exchange is called resolution. Duplex pulled away from one another and rotated 180 degree. Resolvase, ligase, gyrase, and polymerase are the enzymes involved. Vertical cut and horizontal cut. Resolution can occur in either of two ways, only one of which results in an exchange of flanking markers after recombination. The two modes of resolution can be visualized by rotating the duplexes so that no strands cross over each other. In the horizontal mode of resolution, the nicks are made in the same DNA strands that were originally nicked in the parental duplexes. After ligation of the two ends, this produces two duplex molecules with a patch of heteroduplex but no recombination of flanking regions. In contrast, for the vertical mode of resolution, the nicks are made in the other strands, that is, those not nicked in the original parental duplexes. Ligation of these two ends also leaves a patch of heteroduplex but additionally causes recombination of flanking regions. Thank you all.